Shivaprasad after taking his lecture for 1 hour also. Okay, sir. Please, sir. It is over to you, sir.
Thank you, sir, for the kind introduction. Can you all see my slide?
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
O-
Yes.
Okay, so I'm going to talk to you about what we are doing for an hour. I'll start with very simple things and very basic things, maybe in a different perspective. So I named it as more like a plant's perspective. Okay, so I am currently at the National Center for Biological Sciences. My email is here. Let me get one pointer so that I can show you. Okay. So where my lab in NCBS is now 10-year old. I used to work in Arabidopsis and tomato. When I started back, we started with all these model system because what we find in Arabidopsis, we know that whatever work has been done, it cannot be applied to any crop. If anybody has to do something with the to improve the crops, we have to work with these model system, and these are our crops. So I put this mostly for those who are in the now in the beginning stage, more like sometimes I put this for students, but we are all students at different time points. So if we convert Earth's history into 24 hours, what we can notice is that we are really newborns. That's just few seconds older in Earth's history, whatever we are talking about, our innovations, development, etc. But plants are ancient. They are here at least for 6 hours of Earth's time. Bacteria are even longer. They are at least 12 hours. Even the dinosaurs, which stayed for a really long time, they lived only for 1 hour equivalent of Earth's time. So while we have done a lot in the last few seconds, plants also have done a lot. They have stayed here a long time. They have passed through all kinds of climate crises, and they have come sort of successful, or they have been succeeded by other farms, all plants only. Uh, so though they are very ancient, so most of the time what we have it is called as we have plant blindness. And this term was coined by two professors in the University of Missouri who are thinking, why is that nobody considers what they do? They went to their doctor friends, and the doctor friends said, yes, it is because humans, when they evolved, they lost the ability to see things that don't move. So we have sort of blindness for plants, fungi, and to some extent bacteria. All are ancient compared to us. To find out how much of this, how many people have plant blindness, they gave this picture which I put up here. Asked people, what do you see? 999 people said they can see lions. One person said there is acacia and lions. So the percentage is that much, that's 0.01%. But for those who are thinking about fungi, only 1 in 10,000 people can recognize fungi because they just are not moving, they are too small, etc., etc. So one way to avoid this problem or to correct this problem is when children are between 3 to 9 year age, we should give them examples of plants and fungi and also tell them how they are special, what do they do. Otherwise, everybody will get stuck with plant blindness. So those of you are really keen with plants, you must have been exposed to plants by your parents or teachers in that age. Okay, so why is that we have blindness? Why we think plants are not really important or interesting organisms? We think they can't move. A research in my own institute by Professor Sanjay Slane's lab showed that bladderwort has the fastest reversible reaction. I mentioned how fast it is. It is faster than how our neurons work. So it's also a reversible reaction. It's not just spreading like the seeds are thrown out. It is a reversible reaction which can happen in a repetitive manner. So this tumbleweed is one of the fastest. It is actually a group of plants migrated from Russia to US. Those of you are fond of YouTube, you can watch in YouTube. Tumbleweed can move without any fuel, faster than car, even in very difficult terrain. The model has been used to make the rover, which is the unmanned vehicle in moving in Mars. So I'm sure you have seen Mars as craters, but without fuel, this can move. So what about sense? So we think we are with five senses. That is our Panchendriya. So we can sense all these things, and we have necessary mechanism to deal with all these senses, but nothing else. Even these senses are really little faulty. For example, for our brain to know that our tummy is full, it takes 20 minutes. So that's why most of the time we end up eating too much and have a bulge because our brain doesn't know that it is full. So whereas plants, though they are sitting, they have been ancient, so they have made many different kind of sensors. Even the simple plant Arabidopsis has 54 different types of sensor, not just sensing light, which you must be teaching a lot, chemicals, touch, even blowing, even any kind of cue they can sort of sense. And most of these responses are way quicker than animals, including us. So what about brain? The other way we think is sense. Avanige sense idea. So Darwin, who had all kinds of answers for all kinds of questions, even he thought the region which connects roots and shoots, that bulge region, is very similar to brain of lower animals, including, for example, our children. So crow or dogs have equivalent brain of 7-year-old. And what we think is that plants also have somewhat similar kind of a brain. Only thing is it is not connected in one specific place like our brain. So just to highlight this recent experiment to you, so most of the thing I am telling not just a scientific American kind of story. These are all the recent research, which I think it's important we are all aware of this. So glutamate is an amino acid which burns, which is what runs in our brain. And the same amino acid, if we put on Arabidopsis plant, which is a nice model system, you put her here, the response here is measured through GFP signaling. Within 2 minutes, the whole plant knows that you have added glutamate, which is what we use in our brain. Let's go little further. Plants can sense so many things. In a group of plants, if there is a chewing noise by one specific insect, which will be looking like this with the amplitude, it can recognize this insect, how far it is, which plant or how far is the plant that is gets chewed, so that it can make the necessary response already ready, even though the insect is not actually chewing its own body. So from this noise, it can recognize from other insects, other noise or song or wind or any other. This famous paper came in 2014 in a big journal. So what about other senses? So this is the other sense, one more sense. This is tomato leaf. You can see all those tiny hairs. Each of them act like our electric pole. So each of them is tied to calcium signaling. Just for those of you who are not aware, even in humans and animals, most of the time signal passes through that signal calcium signaling cascade, which is shown in color here. Within few seconds, I'm sorry you can't see these numbers, few seconds, GFP and takes the signal, which can be just simple touch, and passes far away. And there is a transcription factor like protein, which can go and tell the other regions somebody has touched or there is something, whatever it is. So that's how sensitive it is. It's really more sensitive than our skin. So what about the other kind of sense? So in our public transport, we don't have people with sense. They want to really jostle around. But look at plants. If you see a region where trees are grown in the same time, they have the canopy shyness. They don't want to intrude with the other neighboring tree. This gap is sometimes 1 or 2 feet width. And in the edges, you will have a different branching pattern. The leaves have a different structure, and nobody has found what exactly is happening here. Just like our www in the internet, they also have a wood wide web. In a group of, let's say there is a forest and one tree gets a phosphate stress, all the trees know that there is a phosphate stress for one tree. They already upregulate the responders like phosphate transporters, even though it is far away. Many times this connection is not just through mycorrhizal hyphae, they also can make sense, make noise. The last year, somewhere end of last year, there were famous papers showing that plants can make noise, which can be heard by smaller animals. And if you keep the right machine, you can know plant can make the noise for specific for drought stress or phosphate stress or some other stress. They make these noises. So everybody, those who are really fond of plants, they know that, plants is not considered as a hot topic. I just want to make one more funny connection here. So we are all warm-blooded. We are pretty proud of ourselves, but 1.5 degree change in temperature, we will be put in ICU. So look at some of the plants. These are plants you're all quite familiar with. And these set of plants, in their sterile male floret, the temperature reaches 50 degree centigrade. And in the night, this can act like a beacon for all the insects because there will be warmth. Warmth means then they will end up pollinating. Even in colder regions, the same mechanism is made to warm the region around it so that cellular constituents, DNA, RNA, protein, they're all intact and the plant can still function. So I put this example for a very specific reason. For those people who are not really caring for basic or fundamental research, the mechanism identified here is this. You need two set of enzymes called uncoupling protein or alternate oxidase to shunt the mitochondria. You're all familiar with those set of complexes in mitochondria, which will generate the heat, finally energy. The same thing can be shunted by these two proteins. If you have this protein, you end up making this high temperature. Somehow humans have lost these enzymes, plants have them, several other organisms have them. Trypanosoma brucei, which causes African sickness, it has only this. So the current vaccines, they target this alternate oxidase, which obviously there is no alternate oxidase activity in humans. So the vaccine works very well. So just see how one can make the connection if the information is correct. So all this wealth came from plants. Still, I think everybody knows how many type of breeds of dogs are there or other hedges, animals are there, but I don't know how many people can name 10 different type of grasses among you. Anybody can name 10 different grasses or sedges? No. So plants are really part of our life so much so that we have forgotten them. This is what I put here is the second longest man-made structure in the world. And this is called Inland Customs Line. And this is second only to, I want this more interactive, so please talk. Second only to?
Great Wall of China, maybe, sir.
Yes, Great Wall of China. So this is the second longest. This is 4,000 kilometers long. And this is where it is. There is only 150 meters of this wall is still left somewhere near Mathura. The rest is all gone. What kind of wall is this? This is simply made with plants. So the person who made this is this guy. Do you remember this person anywhere? Think that my lecture is also a quiz for you. This is A.O. Hume. Any connections?
Yes, sir. This is, I think, Ellen Octavian Hume.
Very good. Yes. So this is the one who started Congress Party, but he was also the revenue inspector. He didn't want the salt-rich region to move the salt to the populous region. So he made this wall so that people cannot move the salt. And when this wall was made, sometimes it was 10 meters wide, and people moved all kinds of thorny plants, including this, everywhere. Now you will find this plant everywhere. It can grow in literally even without water, salty, any condition, it can grow. And this is called Carissa caranda. And I would say many of you might say, I don't know, but you have either consumed it or using it almost every single day. So the cherry which you think in the ice cream is not cherry. It's actually unripe fruit, which is pickled and flavored to give you that feeling that it is cherry, and many other products. I am sure you're all watching what you eat. There will be many products which will start with E303, whatever. These are all stabilizer, enhancer. What are they? They're all coming from these kind of sources. So with that example, I want to say, we all, whether we want it or not, we all continue to depend on plants for food, clothing, medicines, shelter. Even during COVID, first thing everybody wanted to check was whether any plant-derived compound can stop CO2. And they went back to 4.5 lakh compound list, which are already purified and kept to see whether anything can reduce CO2, and many of them did. So what we want to say is we want these climate resilient crops. That's the relevance to your refresher course title. So we all want climate resilience somehow. Our aim is to get higher yield in all seasons, all conditions in a sustainable manner. Although we ourselves don't know what is sustainability, we need 3 cars, we need plastic bottles to carry water, but all the burden should be taken by other forms of life, such as plants. They're already like carrying 2 tons weight for a two-wheeler. Now we want them to carry double of it. So we want further improvement yield, and we want to develop salt-tolerant crops, but we must remember incremental knowledge is not going to be helpful. So let me ask this question: why is that plants make all this? Why do they make all these compounds? So anybody has any idea? Okay, maybe no. So plants make, I'm going to say something which you might laugh, but I think if you go through all the literature, you'll see how it fits or how there is enough literature. They made this to have a nice collaboration with the animals. If you look at the time of when we had the dinosaurs, there were no flowering plants. The dinosaurs lived and died, probably died because they didn't have enough food to eat. Flowering plants were not there. Flower plants came in the last 100 million years. Darwin, who knows everything, he called this abominable mystery because they conquered the earth in literally no time. If you already saw, they came in the last few minutes of flowering plants, and they conquered all other plant forms, and they dominated. In any campus, you will see probably 99% of plants are all flowering plants. So plants tricked animals to if they offer something in the flower that is for spreading them or their genes, they made fruits which can help them with better dissemination. So they conquered that. How is this a collaboration? This is a collaboration because until then animals were coming to the world stage like people in skids come and run away. But once there was food security, then they could stay, and this ultimately led to their evolution, and finally evolution of mammals and us, who are in the last few minutes again. But this interaction was really the key for how the face of the earth changed. And to interact with animals, plants made all these compounds. So there are two problems for plants. Animals put their energy, money on two things. One, on locomotion. Second, on consciousness. That's our brain. So locomotion means you need to control in a central place. That's why we developed a brain. Whereas plants did not do it. They put all their money in chemistry because they were already good in chemistry. They could break water, which we struggle to break even today in lab condition, to make oxygen. They can merely harvest sunlight and make extraordinary chemical molecules, such as the one which we eat or we use all the time. And in this interaction, so they had to ensure that they not just bribe animals, but also somehow control the animals. So they used many different ways to do it. They, of course, provided a lot of carbohydrate and protein, which I mentioned. This is the bribe for the animals. But because they are sitting in one place, they didn't want the animals to come and completely decimate them. They made nicotine. So anybody is smoking in the audience, you don't have to feel shy. Anybody is smoking? Hope not, because nicotine in this tobacco, it can remove the collagen, it can destroy the collagen, and collagen is part of the muscle. Not just for us, all animals, there is no muscle strength if they consume tobacco leaf. And that's precisely one of the way the plant can stop animals eating. I'm sure you know this. Most of the sports people, they never consume tobacco because they can't compete anymore. Caffeine can stop hunger, not just for us, many different animals. Scopolamine at low doses is medicine for Parkinson. Slightly high dose, it induces madness. This plant is Datura. So no plant will be thinking of food when it is mad, right? Cyanides, I don't have to give example, not just for us, many animals, it can stop the life cycle. Flavonols are interesting compounds. So this I put is gooseberry, it tastes. How does it taste?
Initially, it's sour, sir, but when you drink water, it will be sweet.
So it is bitter, then it becomes sweet. There are also flavonols which are sweeter. After that, whatever you eat will be bitter. You have to read some of the Veerappan's, account in the forest. So what are plants doing? They don't want us to consume specific part too much, or they make us eat something else. Photosynthesizers, there are compounds which are more toxic than anything we can make in the lab. Photoprotectants, these will include anthocyanin and flavonoids. They are part of the bribe in flowers and fruits. Then I put set of plants here. These are psychoactive substances. The first one is cannabis. Why do plants make all this? They hardly have much energy in them. So they're thought to make them because somehow animals, especially us, we are fond of altering or improving our consciousness. Even today, you heard we take tablets to improve our memory. So these compounds alter our consciousness somehow for especially us. You must have seen these tiny kids rotating to get visual hallucination. They are very happy, but elders cannot do it. Why do they do it? What kind of enjoyment they get? Because you sort of get better hallucination, and that's the aim of humans. And many thinkers, writers, readers, all kinds of famous people, they are all under the influence of these compounds. So plants make them to alter our consciousness. And some of the best examples are with cannabis itself. There are two species of cannabis, one in Mississippi, where you get a lot of water, so plants can grow a lot, and they make less THC. This is cannabinoid. Whereas the one in Afghanistan, which is a harsh condition, it makes a lot. So what people notice is that the plant eaten by goats in Afghanistan will not find the same plant ever again. So that's basically what it is, and the influence of these plants in our daily whatever we are using, you must be knowing. This also includes partly our spices, which you are too fond of. Okay, so this kind of balance went on nicely because plants could counter everything until the onset of agriculture that happened roughly 10,000 years ago. Here, plants targeted us because we put too much energy on our locomotion. We have complex thoughts. We wiped out literally everything in the earth to pamper few plants. The plants which used us are these plants. These are plants that don't have colorful flowers, but they have carbohydrate. Carbohydrate is an excellent source. Unlike protein, it can be kept forever. You must have read about reports, 2,000-year-old rice grains found. They will be still nutritive. They will still produce ATP. So this was a better option because one can keep these grains for a long time. Our great grandmothers who saw this, they started growing the plants nearby. They started making fences. They started making borders, all kinds of things. Their nomadic nature was lost, and then they started living in one place because of these plants. Now, in this coevolution bargain, where everybody both should be really having some bargain of their own, more grains for us, but dominance in numbers and pampering is for the crops. I don't know whether you get what pampering means. I'm sure you have seen in the fields of cabbage and cauliflower. Have you seen those fields without anybody in it? At least in the daytime, there will be always somebody doing something. So this is pampering. Even if you really provide everything, plants are not going to cope unless you are there. And now even there may be poor houses, but you make better, nicer greenhouses with the even altered humidity, etc., etc. So this is pampering. That's where plants have taken us. So in this sort of coevolution bargain, rice became one of the most abundant macroorganism on earth, maybe along with wheat. So rice is very important for us. Nobody need to say, nobody need to be told again. So the rice that we eat, its ancestor is like this. This is wild rice. This has a long rhizome, makes few leaves, stem is very soft. It is semi-aquatic weed. And it can make only 2-3 grains, and the grains fall off usually in December. That's in the places where I collected such plants. In 10,000 years, we have converted them to such plants which are strong stems and can hold really lot of grain. So it has baffled scientists. How did this rice become this? If anybody still finds it hard to compare, it is like monkey to man. How did we change so much? Now, indica rice that we eat comes from Oryza nivara. It is still found in the edge of lakes in very specific regions. In the peninsula on either coast, you will see, and in the Ghats. And this is prostrate in nature. The Japonica and Javanica rice mostly came from Oryza rubipugan. Both these are still present in India. So what happened during domestication? This is really a puzzling question. Obviously, everybody want to first check the genetic changes, that is changes in the 4 digits ATGC. So you should find some change here, but what happened is very few genetic changes were associated with domestication have been identified. I don't know whether all of you are aware about QTLs. Breeders, when they identify large chunk of region in the genome, they associate with a very specific trait such as yield. In rice, there are 200 yield-related QTLs, and we know only 8 genes when we started this work. That's just 8 genes, though rice is most important crop. So we don't know what exactly caused this domestication-associated change. And landmark papers from Purganan lab in New York City showed that genetic changes are not proportional to phenotypic diversity across all crops. So those who study soybean must be knowing that one handful of seeds from China is the ancestor for all kinds of soybean that we see today. Just from that, you get every color, now every type. So we thought maybe there is role for epigenetics here. I will explain what exactly this I mean. So DNA is ultimate book of recipe. Each organism has it. Each one is packed for kilometer, sometimes meter length of long DNA. This book of recipe, only few of the recipes are copied at a given time. Just like, few recipes which are done every day, you will then make the right dish. Some of those recipes you rarely copy. Let's say it's reserved for the guest or specific condition. So though you have a book of recipe, only a part of it is always converted into dish or protein. So what happens to the remaining one? Or how can one make very specific dish in a given time? That's regulated by epigenetics. In small RNAs, this is largely driven through molecules called small RNAs. These are 21 to 24 nucleotide bits, and they are like from the bits of the copied recipe, but they can cut more, remove the entire copy of recipes, or they can interact with the DNA and cause changes in DNA or histones without changing the digits. So ATGC remains same, but you add very specific tiny chemical modifications to DNA and histone to stop this recipe being read at all. So very one small part of this work got Nobel Prize for Mella and Fire. They did one very tiny experiment. I don't know whether anybody else got Nobel for such small experiment, and they got in no time. You see, they all have black hair, right? Usually people who get Nobel Prize, they'll have white, everything because they have waited for a really long time. Just I'm joking, these people got Nobel Prize in no time. So just to show how important this field is. So the pathway, if I put all the players, looks more complicated. You don't have to remember all the proteins here. But any RNA, this is the copied recipe, can, if it gets converted into double-strand RNA form, DNA is double-stranded. Imagine a double-stranded RNA. It will be cut by a protein called dicer, named by a student who was cutting carrot in her kitchen and thought just like the nice enzyme which can make equal cuts. So, with the nice knife, the protein is now named dicer, the one which can cut both RNA. And these small RNAs can get into protein called organoid. And in the organoid, once they get in, one strand goes out. Now, this is a magic molecule. This ribonucleoprotein complex, that is RNA plus protein complex, can go and do many, many different things. And they can typically go and find anything that has complementarity. The complementarity here is G should pair with C, A should pair with T or U. In that 21, if they find a pair, so pair is like this, the target will be cut exactly between position 10 and 11. Okay, so this is how most plant small RNAs work. Not so similar in animals. Animals, it is mostly translational silencing, where small RNA bind to the 3' untranslated region in the coding sequence. and this repression is reversible under stress. So this pathway has very little relevance for animals because they have different other mechanisms, but for plants, this is a major mechanism. For plants, this is a major mechanism because of this branch of the pathway. Here, certain size small RNAs can go and talk to the DNA and induce methylation of DNA at cytosine basis, and this is called small RNA directed DNA mechanism methylation, and it is unique to plants. The second modification is you can also make de novo fresh histone modification, which is also shared with Saccharomyces pombe. Together, this is called epigenetics or transcriptional silencing. What is very special about this is that once you change and now stop or make the recipe, alter the recipe without changing the sequence, the change can be inherited in the subsequent generation, just like what Lamarck envisioned. Only problem was he used giraffe. He should have used plants' model system. So this mechanism helps plants to cope with all kinds of things. They take the sensors, will be small RNA. Let's say there is a branch of plant which got too much light. Small RNAs responding to will be made. They'll go and alter the epigenome. Different recipe will be read out. And this change will be faithfully passed to the next generation. So I'm spending more time in this slide because it's really important. This is what differentiates plants from us in a way. Our germ cells are decided even before we are born. When we are in mother's womb, they are already made, right? Where is plant's germ cell? So it doesn't know where it is going to make the next flower or seed or next cutting or whatever. So it's going to make, that means it is going to maintain its genome much better than us. Humans have transposons jumping in skin cells and in our brain. That's why we behave maybe differently at different time. But plants, they maintain the genome perfectly because they don't know where the next germ cell will come. And the cue they get will be passed faithfully to the next generation. Okay, so over the last 10 years, we have identified new players in this pathway, such as how small RNAs of microRNA type are made, what happens with specific organelles in rice, which has double the number of organelles than Arabidopsis, what is the effect of this small RNA targeting in plants, how epigenetics works, especially in plants with these players, and what kind of molecular rules are involved. We also work with some other signaling pathways, such as during wounding, glycation-mediated repair, where we identified a new pathway for DNA repair in plants, and so on. So I'm going to stick to some of the work, only two of the work, mostly from Shweta Chenna, as well as from Vivek Harisundar, some of the recent work. So this is how the epigenetic changes look. DNA, if you have this tiny methyl group added to cytosine, it becomes DNA methylation. This is one of the epigenetic mark. One change in the promoter is already enough to stop your recipe being read again. So one cytosine, you know how many bases are there. Promoter can have 2000 digits. One cytosine is already good enough. So whereas histones are this protein drum-like where DNA coils around. The tails of this protein come out, and these are N-terminal tails. They can also go through certain chemical modifications. Again, these can make either the recipe read or cannot be read. Okay, so this is basis of epigenetics. Now, to study if in rice small RNAs contributed to domestication, we collected 400 land races, 2 wild species of rice, and several high-yielding cultivated lines. I'm sure you all know what high-yielding cultivated lines are also called. These are green revolution lines. Land race are those seniors were eating until 1970. These are locally adapted, so lines, but they don't make much yield. So to give an example, high-yielding variety can give up to 5 tons per hectare, whereas landrace can give half ton maximum. So people don't grow this anymore, but they have very important characters, and they are important for religious and many other things. So what we did was we compared genome and epigenome variation, DNA methylation, histone modifications, RNA, small RNA across these lines, selected 12 of them after phenotyping. And I'm going to talk about one such variation that is in the small RNA. So this work was completed by a very hardworking, fantastic PhD student named Shweta Chenna, who is now in Copenhagen. What we found is that typically in all plants, you get this kind of small RNA profile. That's a lot of 21, maybe double the amount of 24. You take any tissue across any plant, this is how you should get. But in Nivara, especially the direct wild relative of indica rice, you got lot more 22, which could not be explained because no plant has very high levels of 22 reported in literature. So if you look into this graph, Nivara has lot of 22. This is just 22. Rufipogan, little bit, but compared to others have very little number. So rice has 12 chromosomes. We wanted to check where are these small RNAs coming from because small RNAs are encoded by the genome. So in chromosome 2, between certain markers, we found a new small RNA. It is a new microRNA because it is not looking like anything people identified before. So if you look at the abundance of this small RNA through next generation sequencing, so it is a technique where you can clone DNA or RNA and sequence in a new platform where the reads will be in millions, if not sometimes billions. And these reads only means if there is more means there is more presence in a given tissue or a cell. So this small RNA reads of 140,000 in wild species, intermediate in landrace, nothing in cultivated species. So just to remind you the scheme, wild species gave rise to high-yielding cultivated lines. In between, there were landraces which are locally adapted intermediates. Now, if you make small RNA northern analysis to check the levels of these after doing NGS sequencing, you can see there is a blotch in Nivara, intermediate levels in landrace, and almost nothing in cultivated lines like Ponni or Pusa Basmati. Now, what are the targets of this microRNA? I mentioned that microRNAs can go and pair with the target and cut between position 10 and 11, and we wanted to check what those are. We have many different tools to do it. What we find is that this small RNA can target enzymes called lactases. So these are key enzymes. They can convert monomers of alcohol into polymers or organic polymers called lignin. Lignin is important because it provides mechanical strength. And this is the second most abundant organic polymer on Earth, after which is the most abundant. All are hungry. Which is the most abundant organic polymer on Earth? So it's cellulose. So it is, after that, second is lignin. Lignin cements vasculature to provide mechanical strength. And it was played a big role when plants moved from aquatic system to land system. So we find these enzymes as the target of the microRNA. Rice has 24 such lactases. Nobody had studied any of them before. And they are quite different from each other, but still microRNA can target all these because microRNA target region is conserved, and this region codes for such a peptide called inlan, and such a peptide is seen only among lactases. So we have many techniques to find which are the authentic targets, which one of them is called degradome, which uses these bits of cut RNA, and we found only 3 lactases are finally expressing in the wild species out of 24 because of this targeting. So this is how it looks. If you look at the expression of lactases, almost nothing, this is with RNA-seq, nothing in wild species, intermediate in landraces, high expression in cultivated lines. Now, if there is less lactase, you would expect less lignin. That's what we see in Nivara compared to cultivated lines. And if you make fluoroglycinol staining in the stems, you get low signal in wild species compared to cultivated species, which make much more lignin. So these proteins are glycoproteins. So we used concanavalin column to pull them down and look for the activity. What we observe is that as expected, low in wild species, high activity and presence in cultivated species. All this suggests that lactase mediated this change might have contributed to domestication associated phenotypes. So this is sort of, we are guessing there is a correlation, very high microRNA in this semi-aquatic weed, which removes lactase, so you get low lignin. Plants are prostrate, they are closed panicles and seed shattering, all connected to lignification too. During domestication, the microRNA is lost. Laches express a lot. So you get high lignin in the tissues, which can support more panicle, grain filling, shatterproofing, etc., etc. So now, which I'm not going to tell is, we know now how this microRNA is reduced. There is a transcription factor which sensed what changed during the domestication of rice. So now the microRNA is gone. So till now, this is a correlation. Unfortunately, most of the research what we do ends up as correlation unless we go and genetically test them. So here, the only way to test is bring this high microRNA into cultivated line as a transgene and check what happens. Can somebody guess what should happen if you bring the high expressing microRNA, put it in the cultivated line, what should happen? So in this microRNA is high, so you got low lignin. The microRNA is low, you got high lignin. If you make the microRNA go up here, you expect low lignification. So when we tried transgenics in 2 different indica lines, so we are one of those very few labs who can make indica rice transgenics through agrobacterium. We got high expressing lines, which made less lignin, and you can see they don't have any mechanical strength. They made a total of 0 seeds. They were all floppy. If you now express lachases in plants, some specific, we tried 3 of them, the plant is much more sturdy and it also made much more yield, suggesting that this is a key for indica rice transformation. How to check this? So we were wondering how come people didn't find. There are millions of Chinese researchers working on rice, especially how they didn't find. So we went back to those 200 yield-related QTLs that all the breeders have come about. They have found over last many 7-8 decades, mapped lachases and microRNA with them. What we find is that 26 of the remaining QTLs mapped to laches and microRNA. So I mentioned that previously we knew only 8 genes. Now we know so many genes which are required for rice yield. And this is how it looks. These are all the genes known for QTLs known for rice yield. These are the known genes which others have found, which include something with the architecture, seed shattering, etc. And this is what we identified in one single study. Many of those QTLs had only lachases. So really it's an important change that happened during indica rice domestication. We got very good comments and lot of coverage, and our paper was for 2.5 months most trending in Plant Cell, which is a premier journal for plants. And in the country, just a comparison, not more than 10 Plant Cell papers have come from our country so far. So one more student was looking at the flower color or fruit color in the lab. And how it started is we have smaller data sets from really 120 different plants. And suddenly we found one set of microRNA very highly expressing in gray. And this targeted a gene which is a type of MIP transcription factor, which expresses only in reproductive tissues. So we connected this to the pigmentation, and pigmentation in plants comes from anthocyanin. That's what you see here. So can somebody tell me what this first plant is? This flower. Can somebody say what that flower is?
Lily.
Not the second one, first one.
Look like a cotton flower.
So this is a tulip called Semper Augustus, and this is the one which caused tulipomania. Literally, Dutch became crazy because they were very fond of this coloration, of specific coloration of anthocyanin. So Dutch really sold their land just to get few bulbs, and this is literally the start of the share market 400 years ago. So we all like colors. So what we did is once we called all the high school students and asked them to choose among 20 fruits, we kept only 3 colored. We found somehow all colorless fruits or green fruits, and everybody wanted colored fruits. So it's in our genes. Somehow we want more colored fruit. Not just us, many animals, including birds, also like colored fruits. So that's why we think all these compounds are really helpful for us, for our health. We pay a lot of money, but our grandmother was right. You can't use this. It's not going to be, cannot be used because you will be breaking you have to break the vacuole in our system rather than by making a juice. But whatever that is, for plants, these compounds help with attracting pollinators. If they're in flower or if they're in fruit, they help with dispersal. So when we studied this interaction, what we found was we used the grape. We used 3 different grape varieties to do whole genome transcriptome, look at RNA, look at metabolome, look at small RNA. The small RNA has to be very high only when there is color. So you make anthocyanin. So the reason is when we started becoming very fond, grape is one of the, I think probably the oldest domesticated fruit. The number of MIBs became many more because we were looking for more color or flavor. And there was evolution of MIB repressor. If these are cut by the microRNAs, you get color. If they are not, if these are allowed to express, then you get flavonols such as in Dilkush. As the name suggests, this is good for heart because it makes flavonols. So anthocyanins are these. They are again very important for health. That's why we are fond of them. And anybody in Bangalore, please don't be too proud that this is made in Bangalore. This is a Iranian variety and started grown in Bangalore, so people started calling Bangalore blue. Nothing to do with Bangalore. So using this study, what we find is that you need a very specific set of regulators to make fruit. After our paper came, now there are more than 25 papers in different crops, including strawberry, apple, bengal, even potato. If you need anthocyanin, you need this interaction, and that's what we had found. This MIB repressor we found is the new class of transcription factor. We call it as MIB114 class. Okay. So fine. So over the last few years, we found so many interactions, connections to small RNA and yield-related traits. It's mostly small RNA and epigenetics. So we started wondering how this works in plants. But because I stay in, first let me say this, because I work in a institute where everybody else is working on animals, they obviously asked how does that work in animals. Animals also, there is a strong connection to small RNA because myostatin mutant is the one which makes a lot of muscle, and it is called double muscle phenotype involved in many of our domestic animals. And this happened if you remove myostatin by having a microRNA mutation. Now the experiments were conducted in Yale and they made a patent, but the information somehow leaked that if you reduce myostatin, you get double muscle phenotype. And there are people who use this to make double muscles. So if you next time look at this bodybuilder, you have to doubt whether the person really pushed the iron or took myostatin inhibitors. Okay, so we find so many interactions of small RNA and epigenetics in yield-related trait. We wondered what is happening in plants. So if you look at plants, I already mentioned a couple of points. Plants have to maintain their genome throughout without a lapse any place. Plus, they have many innovations. They can make next generation from literally sometimes leaf, sometimes root, tubers, of course, flower. So everything is nicely maintained. The genome is really well maintained. And plus, they have tissues such as triploid endosperm, which is a polyploid tissue within them, which is next made to help the embryo which is diploid. So all these interactions suggest that even here, there is a lot of epigenetic and small RNA-mediated processes, all suggesting that epigenetics plays a role, but why? So, or how? In plants, what people have identified is that there are extra polymerases. All other organisms have pol 1, 2, 3 to make mRNA. Plants have pol 4 and 5. These are the ones which transcribe literally every part of the genome, including where we think we can't read the recipe. And all they make is small RNA. And there is a reversible chain. That means sometimes you can read the recipe, sometimes you shut down. And this depends on the environmental condition or the stress they go through. And many of these changes, they are heritable. That's really why plants have managed to stay such a long time. All they need is space. If they are given space, they are going to manage all kinds of changes that we put on them. I don't know, there are recent reports suggesting that because there are less number of bees for pollination. Many plants have already started becoming compatible. That means they have either the filament smaller or stigma now maturing quickly so that they can do self-pollination instead of waiting for the insect. So they're pretty good in managing their own, except as long as we give them space. Many of these epigenetics variants are very well known, including the ones which Barbara McClintock has studied. I'll give one example here. This is a epigenetic mutant where there is no change in the DNA sequence. But before this gene, a MIB 2 kb upstream, couple of cytosins are methylated in this. So now the fruit never ripes. And if you take the seeds from this again is not going to ripe. All what is happening is couple of cytosine methylation, which is heritable in these crops. So if you see many of those tomatoes which are not really ripening well, or they can stay longer, this mutation is involved. This is a epigenetic mutation, no change in the DNA sequence. So something happens also in animals. For example, ants, minor and major worker, there is only histone modification. Same thing also happens in honeybee, where royal jelly has specific histone modifier inhibitors, so that suddenly after eating royal jelly, somebody can become a queen. Okay, so but in plants, this organization is completely different from animals. So I just want to point out this is how the nucleus looks. How much time do I have?
So still for 30 minutes, sir.
I have another 30 minutes.
Yes, sir. Approximately 20 to 25 minutes, sir. 25 minutes.
Okay, very good. I will, at this juncture, I want to see whether you have any question, because otherwise it will become monotonous.
Yes, sir. Yes, sir. Okay, sir.
Do you have any questions?
Participants.
Sir, you said the fruits are not going to ripen, or they are not going to become, their outer wall is not going to change its color. It looks like they are unripened itself. Is it?
Yeah.
Hello, am I audible?
Yes, you are right.
Yeah. That is due to this microRNAs, or else some other pigments, the genes.
No, this is one cytosine methylation in this gene, which I showed here. Here there is no small RNA. Okay, actually there are small RNA. This region makes some specific small RNA, but that's happening because of this DNA methylation change 2 kb upstream of ATG, very far from ATG. So you can see such a regulation is recorded only from plants. And this is already enough to either keep so here methylation means there is no expression of the gene. You get green fruits which are colorless, never ripe. So what I wanted to say is you can find such variation which are called epimutants really lot in plants compared to any other organism, because their regulation is mostly through epigenetics. They can so here there is a trait change, right? You either ripe or don't ripe. That's simply from one epigenetic change. So there are thousands of such changes in plants. So some of the most craziest ones are called paramutation, which is like a contagious disease. So once you have a paramutant cross to anything, that phenotype will be passed. There is no 3:1. It's all offsprings will carry that phenotype. And this is called paramutation. You can read all about them, the best examples from maize. So all these are epigenetics and small RNA derived processes in plants.
So another question. For example, you're showing the tomatoes here. Is it going to affect the lycopene formation also?
Here it didn't, there was some change, but people then bred it to some other varieties to get the ones which are really good. And these are not transgene, just breeding of the epimutant. So they did get the right tomatoes. Many of those you find in our market also have this some methylation in the cytosine compared to, let's say, the wild tomato like Solanum pimpinellifolium. So compared to that.
Thank you, sir.
Okay. Any other question?
Sir, you have said that co-evolution regarding co-evolution bargain.
Yeah.
Sir,
Yes, yes.
and that led to more grains. That led to more grains. So, in case of rice and all the monocotyledonous rice, wheat and oats, and these are mostly wind pollinated, sir. So they are not dependent on the animals.
No, they are not dependent on they didn't make us to do something for pollination. So they, of course, they don't have much to, they don't even have colorful flowers. So they don't depend on animal for pollination. What they dependent on us is spread and pamper them, give them more water, grow them in a nice fashion so that their number go. So in a way you can also, so just like we want to say we did those, but plants can also say we did those. We only made you grow us more because we are able to provide the right grain. So there is somebody called Mike Pollan who has come with this theory. Actually, this looks more correct than what just humans think. So the collective information suggests that other organisms also manipulate us. And this is one such example. So in that bargain, they have become much more successful than us. Of course, we have huge population, but we are going to probably have other trouble, but plants are going to be fine.
Yes. Thank you, sir.
Okay, I'll go. Maybe you can ask questions later. So this in these cells, you can see each nuclei. You have some regions which will never be read, even when guests come. These are called heterochromatic region. So there are two types of heterochromatin. One is constitutive heterochromatin, means those recipes are, imagine they are forgotten. They cannot be read, and this is called constitutive heterochromatin. There is a very specific chemical modification in the histone called INH3, histone 3, lysine 9 methylation. There is one more called facultative heterochromatin, which is like the where you can have some genes coming up like a recipes which are read when guests come. Okay, so these are H3K27, lysine 27 trimethylation. Okay, so rest of the region is typically open for with marks, other marks which are for open chromatin. This organization in each nuclei is maintained by histone modification. I spoke about it. DNA methylation, I mentioned that. Small RNAs, they somehow manage this. So we wanted to see how this organization helps plants to cope and make changes to their phenotype in a heritable fashion. So in plants, you have pole 4 and 5. They read everywhere and make small RNAs. These induce DNA methylation. And such polymerases, they have been thought to help with the plants' colonization, literally that abominable mystery, because all plants have polymerase 4. Pole 5 came only among flowering plants. Now, only last year we found pole 6, which we don't know what it does. It's only in poaceae members. So if you know, poaceae really evolves very, very fast. So if you look at this pole 4 in plants such as Arabidopsis, which is the most studied plant, but here this organization of genes and transposons are there all kept separate. And obviously, you know, this is not a crop. But if you look at any crop, transposons and genes come next to each other. Just to give an example, Arabidopsis has only 3 genes with their transposon in the promoter. In rice, it is 28,000. So that's why repeats transposons really play a big role in the phenotype of crop plants. Now you can see this very well here. In Arabidopsis, if you remove this polymerase 4, nothing happens to the plant. This is what people have recorded. Though there will be reduction in the small RNA, reduction in DNA methylation, nothing happens because it's a nicely organized genome. In rice, when we mutated pole 4, you can see already the panicle is completely different. There is sterility issue. The most pollens are gone bad. At the molecular level, small RNAs from transposons are gone in the mutants and so on. So to some extent, it matches what we see in Arabidopsis, but phenotype completely different. There is no yield here. Now, we made a complemented line. That means in the knockout background, put back the full length gene, and now the small RNAs recover to wild type levels. So this is called genetic complementation. Now, if you look at these small RNAs, we got this dependent small RNA which go in small RNA pol4 mutant. But what we find is that we got a new set of small RNA called pol4 suppressed small RNA. This is the first such report. You can see in some regions, so here is the centromere, you get loss of small RNA, these are pol4 dependent small. But you get new small RNAs which are coming only in the mutant. As you can see in northerns, in both rice and Arabidopsis, they suddenly have come up in different region, which you term it as pol4 suppressed small RNA. We wanted to know how this happens. And what we find is that in plants, in rice, if you mutate pol4, this facultative heterochromatin region really comes up much more. And this region now becomes a pol2 substrate. And pol2 then goes and targets the small RNA coming from pol2 will go and target all the reproductive structure related genes. So that's a strong maintenance of chromatin boundaries in plants, and that's done by pol4. If you mess with any of these, there is no yield. So everything goes to the reproductive structure. All these targets, what we find misregulated gene, they're all reproductive structure associated genes. So no such chromatin boundary regulator has been identified in plants. So we are quite happy about this finding. So what we just I mentioned is you need to maintain this here, which is not happening in animals properly, or we don't know. But this maintainer is polymerase 4, which tells which recipe should be read and which not in a very precise way in a given tissue. So in the last few years, we found you can also have all these combined effect by the presence of what is called as histone variant. So in next 5 minutes, I'm going to tell you what we find with the histone variant. Histones, obviously, these are the octamers. This is called nucleosome when you have DNA wrapped around. Tails are outside. These tails get the post-translational modification. Many lies in histone 3. They get modification. Depending on the modification, either the recipe can be read or not. Either the gene is shut, silenced, or not silenced. In lysine 5 position in H4 is an active mark for all kinds of stress regulation. So this is how our histones look, both plants and animals. You have the DNA wrapped around, tails are outside, which will get different modification. Then they can help with this condensation. So what happened is, because histones are so important, there are many paralogs of histone variants, histones evolved, and these are called histone variants. And unlike canonical histones, which should be decorating all throughout the chromosome, they decorate specific places and they go very specific things they can do. These variants are well known in H2A, H2B, and H3, but none in H4. So what do? When they are present, they'll change that amino acid in the right place. Here, let's say there is a lysine 27, the variant will not have lysine, they'll have something else. So you can't have that PTM, or they'll have another amino acid which will change, give a new dimension to the chromatin. So variant became very important. These work on top of everything. You have enzymes, transcription factors, histone modifiers, chromatin regulators. On top, you will have histone variants for any given change in any organism. So how do they work? If you have a canonical nucleosome, if the system incorporates variant, now the nucleosome core particle positioning will be altered. This is like making makan, right? You do this, right? Same thing can happen with DNA, or you can have flexibility with DNA, how much it is binding or not, or you can simply keep or remove a nucleosome core particle. So we found a new first histone H4 variant last year, and this is the canonical. This is the DNA binding region. This is where the tail, which will have post-translational modification, all shown with colored round balls. But in the variant, you can see there is a completely different set of amino acids only in that tail, but nowhere else. Such a variant, if you look at all the organisms, including human, nowhere else except only in Oryza members. And Oryza has 11 copies of this H4. 10 are identical, every amino acid, only one is variable. And if you fuse GFP to the promoter, it went nicely to the nucleus, and expression-wise expressed very well compared to canonical, except in panicle tissues where it is completely absent. Okay, expressed nicely in seedling stage and in embryo and endosperm, but not in the panicle stage. So the sequence of this variant is conserved across 3001 rice genomes. So to study this further, we generated an antibody very specific to this variant and checked in the where it goes in the nucleus. It went mostly to this region, which are pericentromeric region, which will be mostly silenced region, where you have RDNA repeats, etc. But also 1/3 maybe to the genic regions. So this is non-genic region, silenced region. This is genic region. When we did a chip-seq, that is, cut the whole genome into bits, put this protein, pull it down to see where it binds in the genome. So this is called chromatin immunoprecipitation. We found 1/3 of the regions are intergenic, 2/3 are intergenic, 1/3 is genic region. The region where it binds are only 3,000 regions in the genome. Roughly 3,050 nucleosomes among millions of nucleosomes, and peaks are very sharp, unlike many others that are being studied, and it mostly colocalized in most areas to the repressed mark, that is K9 mark. Now this is what we did next. We purified each of these protein at 2A, at 3A, at 2AB, and the variant, made the tetramer in the lab. We made then added dimer. We can do this with serial dialysis. Then add the DNA called Widom DNA to wrap around and make canonical and histone variant containing nucleosome core particle. So this was done first time. So why it is first time? Because in plants expressing these are very hard. Second, at 2A and 2B have 30 extra amino acids, unlike animals. So if you look at human nucleosome core particle in the gel, runs faster because it is smaller compared to both canonical and variant containing nucleosomes. So with this, we got the first chromatin structure for a plant chromatin. If anybody asks how does the plant chromatin looks, you can say maybe this is how it looks. So this is in the side view. This is for canonical. This is for variant. You may notice this change here. Canonical, the region where this is DNA, gray is DNA. H3 and H4, they are not well resolved. Whereas in the variant, it is very well resolved. So it's basically much more compact in variant compared to canonical. For strange reasons, this is how human nucleosome looks, more compact, and all plants have variation. We think maybe because it has the more interaction with the chromatin remodeler, unlike in animals. We found the amino acid variation that can result in this change. To study this further, we use these NCPs, chop them with the aminase, the bits which are not bound to DNA. We got them sequenced with deep sequencing with the NGS. What we find is that variant can bind longer DNA compared to canonical. So you all know that 147 binding, right? So it can bind slightly longer DNA, but the entry, so this is DNA entry site, this is a exit site, more binding in variant in entry and exit sites compared to canonical. So there is a reason why this can happen. Surface charge of variant containing is more positive compared to canonical. So there is more charge. So DNA can bind better because DNA is negatively charged. So when Vivek, who is the student who did this, he was running these complexes in the gel. He found that the variant is much smaller compared to canonical. So now there are two things. It can bind longer DNA, make more compact, so it also runs smaller in the gel. How exactly this mattered to the intramolecular interaction? We took these tails, fused with biotin, which can be used as a bait, and did peptide pull down assay. What we find is that variant cannot bind to the nucleosome core particle. It can better bind to octamer. So there is just the tail difference can change how this can interact with the nucleosomes. So one of the well-known, only one known feature for variant in plants is there is thermostability. Typically, it should be around 88 degree it can tolerate, but variant was 5 degree lesser, so it was dissociating much faster with this fluorescent assay. If you make that single amino acid variation, 50% was recovered. So most of these features are quite novel for any variant, including animal one. So the stability of nucleosome is what makes chromatin remodelers, modifiers to come and recognize and change the features. So when you have a variant, you can get two things. It can be homotypic. Out of 8, you can have both same, or you can have one variant, one canonical within that. So we wanted to check which of this is happening. We fused with the head-to-A, we fused with his and started doing by providing the molar excess of another. What we find is that it acts only as a homotypic nucleosome, not with heterotypic nucleosome. Now, how does this all matter? We have to take the genetic approach. We made a CRISPR knockout. That means you can edit certain bases so that the gene is completely now removed. And we went all the way to TIFR and removed the Cas9 so that we don't have effect from remaining Cas9 and the guide RNA. So this is what is needed when somebody says they are going to do editing. They have to remove the Cas9 because Cas9 otherwise will give trouble. So this is what is approved by the government. You can make edited line without transgene. So it is then like you use transgenesis but get edited line, which is approved last year. So we picked single copy transgenes and started checking what looks different. Two striking things. One, plant is already stressed. Okay, so the plants are smaller, root is not changed, which is the major change in the knockout line. KO means knockout where you don't have variant anymore. So we also saw some changes in the seed, but I am not going to the detail. We did a transcriptome in these two stages. What we surprisingly found is that in the knockout, you get a lot of this changing, which mimic salt stress, but nothing else. So this is a principal component analysis where we used all other GEO datasets across whomever has done anything, compared to all the differentially expressing gene that we found in knockout. And very surprisingly, salt stress transcriptome. That means when plant is put in salt and what changed in the RNA level, recipe level, is mimicking what we see in knockout. Not even osmotic stress, not even drought stress. People think these are all connected. They are connected like 10 genes, but not for thousands of genes which go up and down in a given stress. So the genes in knockout are already up even without salt stress. So that's what we find. And if you make a regression curve, so here is a scatter plot where you compare differentially expressing gene in knockout with the salt stress, everything is perfectly matching with each other, like superimposing. Only 5% genes behave differently. They have completely different signature, but 95% of all genes match. That's why if you now put the plant in stress, it's not going to change anything. It is behaving because it's already behaving like in stress. So what is happening in the stress? In the under stress, there is more protein going to the nucleus. Now there are few thousand extra peaks which are now wider than what is found, and it went mostly to the gene-rich regions from the silent region. And in those regions, it activated a mark called H4 lysine 5, which I mentioned. So this is a typical stress-related mark. If any H4 is brought in, especially during cell cycle, this will come preloaded with the active mark. What we see is that among the genes that are changing, this comes only if you have knockout. For example, in the knockout line, H4 signal is completely lost because this cannot come in unless you have the variant. So basically, this is what we find. Without any stress, plant has variant which is bookmarking right regions that should go up. Only if this is there, preloaded active mark will come and POL2 will now activate genes that are required for salt stress. We also tried putting these plants back. Now it will go back. There is no more stress. So why this is happening in plant like rice? I showed this slide before. Rice has evolved in this semi-aquatic system. We always worry about water, but we should also worry about the salt. When the water is low, salt goes up. So rice, especially rice, has a very nice way of mediating these changes across in salt level, whether it's in natural condition in the lakes or in our agriculture system. That's what we find here. So people already knew that certain transcription factors has to go up under stress, and these are the cue from salt stress. But how this happens, nobody knew. And we now know H4 variant is the one which can activate all these genes. So I put this mostly as a fun. Do I have 2 minutes? I think I'm coming to 1 o'clock. Do I have 1 or 2 minutes, or I shall?
Yes, sir. Yes, sir. You have 2 minutes. No problem.
Okay, that's just like, thank you. Just like the mobile phone, we are in the 4G stage. This is called design breeding. It's not just biotechnology, lot of editing we have to make by big data, something like what I showed with the rice and grape. Only with this, we are probably going to match the growing needs of the food. But this happens only if you develop climate resistant crops. And developing climate resilient crops is not easy. We have 2 such crops, but it will take many years by the time the approvals come. But climate changes every year, right? Every year, you know what is happening. So in the country, we have exhausted growing area. We have huge, all these are what is happening now. We are importing a lot of food grains, whether you read in the newspapers or not. For example, pulses come from Canada and Australia. But by 20, we will be having no matter who says what, we will have a tough time because we are going to have much more problems with the existing crop area, especially water scarcity. What we can do? We have to develop climate resistant crops. It needs a lot of expertise and a lot of hard data. But what everybody can do is you can have crop diversification, better use of water, and sort of some changes in our lifestyle. So this is what we eat now. This needs 4,000 liters of water. And this is what we can eat. All these are present. They don't need much input. This needs 400-liter water only. And this is much more nutrients. I'm sure you're probably not happy to see ragi, but ragi is such a amazing crop. Only thing is we can't digest easily. So unless we have to come with the innovation to cook it properly. The closest relative is called TEF. We are importing TEF to feed our marathon runners, and TEF evolved in Ethiopia and Kenya. Who wins gold medal in Olympics? It's them, right? They might be thin, but they have the right resilience to go through marathon. Our ragi is not very different. We have many grains which are not very different. Only thing is that our research is just on the surface. We haven't really found a way to use them. And most biggest problem for us, for all of us, is the misinformation, just like which oil to consume. I'm sure you all read about apple a day keeps doctor away. Do you think so? It's a big lie. It's a 100-year-old lie. If you eat apple every day, you might get diabetes because apple growing regions in US, they were making alcoholic drinks. And then suddenly church said, there is prohibition law. You can't drink anymore. All the growers come with this slogan so that people start eating raw fruit. And that's what is kept even today. And I can say just from based on the Wikipedia data, guava, our poor guava is 20 times more nutritive than apple. So most of the information what we find is really rubbish unless you have the appetite for the right information. With that, I thank lab members. So we call ourselves EpiPepis, and the structure we had to we did in our own facility, but we didn't have the right carbon grid, so we collaborated with Jayaprakash. These are my source of inspiration. David is the one who discovered small RNA. Weltham is a famous plant biotechnologist from the country. He has trained 643 people. I am one of them. And thanks to all the funding agencies, and thanks for listening. I would like to thank Dr. Rajkumar for the invitation so that I could share all this with you. Thank you.
Thank you, sir, for your nice presentation. I request